Welcome to Brain Ponderings, your window into the world of neuroscience research with conversations with scientists at the forefront of brain research, neuroscience research, and related areas. Today, the topic is regulation of food intake and energy metabolism. So hunger, satiety, and these interesting connections between circuits in the brain and how your body handles energy coming from food in reciprocal manners. And I have a world's expert on this, Henning Fenselau. He's um, he's at the Max Planck Institute for Metabolism Research and the Polyclinic for Endocrinology, Diabetes, and Preventive Medicine, and the University of Cologne Excellence Cluster for Aging and Age-Associated Diseases. So that's a mouthful. Henning, you're in Cologne, I understand.
Yes, I am. Cologne, West Germany. Yes, Germany.
And yeah, West Germany now. But
Yeah.
I mean, West Germany then, Germany now.
No, yeah, I'm referring to in the west of Germany, not
Yeah,
not West Germany.
yeah,
Yes,
okay.
certainly. To clarify that, sorry.
And okay, so where are you from originally?
I was born in Bonn, so very close by.
Oh,
Yeah. So and then did some time there and here. So in Vienna, I did my PhD and my master's, and then went to Boston, where I did my postdoc, and then came back to Germany, uh, to Cologne. Now 7.5, 8 years ago.
and how soon in your life did you decide you wanted to be a scientist?
I think it's difficult to say when you want to
Yeah.
be a scientist before you know what a scientist is, but I was always very interested in understanding feeding behavior, food intake. Uh, Probably because I was just interested myself. Why are we eating? What are we eating? What makes us eat what? And yeah, the one led to another, and then I became more and more interested in like studying this, and this is how I became scientist, neuroscientist.
And okay, so what was your early work? So you mentioned you were at in Vienna and then postdoc at Harvard with Brad Lowell, who's a very well-known, well, I guess you'd say neuroendocrinologist. Um,
Yeah,
yeah.
yeah. So I originally studied nutritional science. So I studied nutritional science, studying in Bonn. Then I moved to Vienna, where I finished my studies. And I did work doing my master's or my master thesis on gut uh, absorption, digestion of peptides.
Hmm.
And then I moved very far away from this because there were different reasons, personal but also scientific reasons that let me continue studying more neuroscience and really dive deep into sensory neurons, synaptic plasticity. And then I learned about, after my PhD, where performed a lot of electrophysiology for, I learned about the fantastic work from Brad, and this is where I contacted him and then joined his lab for a postdoc. And that was the changer, where I like learned about how important the brain is and how complex it is. And studying this really fascinated me, and this still fascinates me.
Now that of course, obesity is a big problem. Certainly here in the United States, it's a major burden on the healthcare system, and there's a lot of reasons for it. One is, there's companies that make highly palatable food. It's addictive in a sense. And then once someone gets addicted to overeating, these systems that regulate hunger get a little out of whack, and the individuals perceive that they're really hungry, even though you clearly have a lot of energy stores available. So the early work on control of feeding, and still a lot of the major work focuses on a brain region called the hypothalamus. Can you talk about why that's so important?
Yeah, I think as you were referring to, right, there was early work. I mean, there were work in rodents, but also from humans, right? There were patients that had lesions in the hypothalamus and they became hyperphagic, so they couldn't control their satiety, their hunger anymore, and they just were eating, right? Eating. And so really overeating. And so that then led to this idea that you have these homeostats somewhere in the brain, and that must be in the hypothalamus. And obviously, I mean, our way of doing research in the brain has changed dramatically over the last 20 years. We now have tools to monitor, manipulate, and look really in depth what are all the cell types. And um, so the hypothalamus became, yeah, already 30, 40 years, it became clear it's really this complex region that contains so many cell types that are involved in the control of feeding behavior. Because when you manipulate them or when you ablate them, you see these very, very strong phenotypes that overeating but also undereating can occur. And that then really placed the hypothalamus at the center one of the brain areas that is so critical for the control of food intake.
Yeah, my background is my early work was really neuroendocrinology because I was studying the master's degree. I was studying regulation of cortisol release from adrenal gland cells, cortical cells, and then my PhD was in studying a neuroendocrine system that controls molting in crabs, which involves a steroid too. And so at that time, I wasn't really up that much on the regulation of feeding, so I just thought a hypothalamus is a it's a region of the brain where there's some, usually a peptide, released into the blood, and then it goes to some endocrine gland and causes that gland to secrete a hormone. So in the case of the hypothalamic pituitary adrenal axis, it's ACTH, which causes cortisol release. And then, but the feeding circuit is much more complicated than that.
Yes, absolutely. I think this is how people studied it until it became clear that these cells, that not everything that the hypothalamus is neuroendocrine, right? But that it does much more, and
Yeah.
it has this capacity, right, to like read out signals from within the body. So hormones, particularly, right? I think leptin is one of the most important ones for reading out the energy state, the signal coming from adipose tissue. But then it also communicates signals from that it's generating from like these signals to other brain areas, but also integrates information from others. And we're learning more and more about this, obviously, with all the tools that we have now. Yeah, photometry, whatever, you name it. And that I mean, the more we learn, I have the feeling the more complexity we realize there is in the hypothalamus. And
Yeah,
it's
and
much more than just a
it's so conserved in evolution. I mean, all mammals have hypothalamus, and yeah, birds too. And so but when a human brain's so big, and then when you like look at the size of the hypothalamus in relation to the whole brain, it seems tiny. And that, if you just kind of think of this, I guess in a superficial way, you say, well, it's so small. How come be it so important? But it is. And all these other circuits higher up, their interactions with the hypothalamus. So I mentioned addiction, right? So that involves, whatever the dopaminergic neural circuits and so on. And hunger is critical for survival and stimulating feeding behavior, right? So you go and find food, kind of a pressure there. So there's other circuits involved in motivation, I guess, and even cognition that are interacting with these circuits in the hypothalamus.
Yes, absolutely. And I think, I mean, it must, right? Like the hypothalamus by itself cannot do anything. It must communicate to
Yeah.
all these other structures to drive motor output, to drive learning. And we don't know, right? I mean, hypothalamus is communicating heavily with the dopaminergic pathways, and can do this in a state-dependent manner, right? Like you should prioritize feeding if there is no energy in the body, right? Drive hunger, to make this the most important, because if you don't eat, then you can't survive. And so this is, I think, from a hypothalamic perspective, you can always say it's the maybe the most important brain region. Maybe some other people working on other brain areas would challenge that view, but I think it kind of is, because if you, I mean, do these lesions or see manipulations to it, they cause such massive changes, right? And I think it makes total sense for the survival of the species. It is so critical to have a sufficient amount of the food, consumed. And the hypothalamus is also driving thirst, right? So drinking, which
Ah.
is as important, sleep. All of these major homeostatic functions are driven by specific populations within the hypothalamus and their pathways that are ultimately controlling all of the cells in the brain.
All right, so the hypothalamus, there's different collections of neurons, nuclei, we call them a collection of neurons. And can you talk about some of the key regions involved in hunger and satiety?
Yes, so one region that has become particularly or can, was shown to be so critical for feeding behavior is the arcuate nucleus of the hypothalamus. So the arcuate nucleus is very really at the bottom of the hypothalamus, and it's directly next to the third ventricle and the median eminence, and that makes it also unique in its way, because any signals that are in the circulating system can
Mhm.
more easily penetrate into the arcuate nucleus. And so even though the arcuate nucleus is really tiny, it's a very small nucleus, it contains some of the most important neurons that are controlling hunger and satiety, namely the cells often referred to as AGP cells, so agouti-related peptide expressing, and those that are controlling satiety, those are the ones that are pro-opiomelanocortin, or in short, POMC cells.
Okay, and so those two different types of neurons must be connected to each other. How do they communicate with each other? You do a lot of work on synaptic communication, right? And that involves neurotransmitters or neuropeptides, which are, I don't know, I don't know if you should even be consider them as a separate class, right? Because they're released at a synapse and act on a postsynaptic cell. But anyway, these different signals at synapses, what are some of the key transmitters at the synapses?
Yeah, so what we know about AGP neurons, let's start with those. So, because they're very famous, I would say, right? These are the often referred to hunger neurons. And these neurons are inhibitory, so they're GABAergic, and in addition to this fast-acting neurotransmitter, they also produce two neuropeptides that are inhibitory, which is neuropeptide Y and AGAP, which they're make them very unique because that peptide is in the brain only expressed in these cells. And so these three together work to inhibit the downstream partners via, obviously GABA A receptors or GABA B receptors, but then also NPY receptors and melanocortin receptors, particularly the melanocortin 4 receptor. And so via the release, they can inhibit the downstream neurons. And
Okay.
they also communicate to POMC neurons, so these nearby cells. So if you would like to, I mean, you can always refer to them as like the yin and yang. So the hunger neurons are these AGP, the yin, and then the POMC are the yang that are producing satiety. Um, so obviously, if you want hunger to be produced, you want the satiety to be inhibited. So it makes total sense that what was found is that AGP cells communicate to POMC cells and silence them, so to reduce satiety when in a state of hunger. Um, and so the POMC cells, they appear to be primarily neuropeptidergic. And they're extremely famous for their alpha MSH, that is a neuropeptide that they produce, which acts as an agonist on the melanocortin 4 receptor. So it does the opposite to AGP, and also via this can work in an antagonistic manner. But POMC cells are also a little uh, not well studied for all the other neuropeptides that they're producing, and it's not fully understood what they do. And this is something we're actually very interested in.
What's the history? I don't know that. What's the history on? So MSH is melanocyte stimulating hormone, right? So melanocytes
Alpha.
are like control pigment in the skin. Is it?
Exactly.
Yeah.
So this is why they were also like found first. So the melanocortin receptors were formed first in peripheral organs, and then it was identified that there are other melanocortin receptors. So the melanocortin 1 receptor, I think, is the one that is in the periphery and also for pigmentation. But then there were the identification that there are more melanocortin receptors, and particularly the melanocortin 4 receptor became very prominent because that is expressed in the brain and is causing obesity if it's lacking. So also in humans, deficiency of the receptor causes obesity. And that really brought this melanocortin pathway on the map, that lack of melanocortin 4 receptor, but also lack of POMC. So humans that have deficiency of POMC or also knock out mice, they also develop obesity because they don't have this satiety signaling functional property anymore. So they have no control over feeding because they have a lack of satiety signaling.
And then if you right, and then what are there any well, I guess you that would be selected strongly against an evolution. Are there any mutations that cause uh, constant satiety so the individual starves? Or can you do that experimentally, I guess, with a lesion or some other manipulation?
So that was done experimentally. I think evolutionary speaking, it makes more sense that you have always hunger, right? Because that
Yeah, yeah,
obviously drives animals to eat more.
yeah.
It's actually interesting that there are data demonstrating that uh, dogs that learn better seem to have a higher preference for melanocortin receptor 4 deficiency or POMC deficiency because they're more motivated to get food.
Interesting.
And I think this is very interesting from an evolutionary perspective, right? It makes total sense that those that are more driven to get food are more likely to survive. And so there might be a trace why the deficiency of the receptor or less efficiency of it might, evolutionary speaking, make sense.
Okay.
But
And
coming back to, I mean, the other way around, right? If you, what has been done experimentally, if you um, ablate the AGP, so the hunger neurons in mice, in adult mice, they stop eating. So it's very severely affecting them and make them, the reducing the hunger drive, at least under control conditions. So that was shown very nicely by Richard Palmer. He did fantastic experiments there.
all right. So what in your let's talk about your laboratory itself and some of your colleagues there, your trainees that are working and uh, so what kind of methods you do most of your work in mice, is that right? And
Yes.
how do you how do you look at the function of these different neurons in the hypothalamus? And then, lesions are kind of crude, but, very important in the early work in identifying this. How do you manipulate the activity of the neurons? How do you, I guess you use pharmacological approaches to try to identify what transmitters are involved. Can you kind of talk about, how you've got it organized so that you can get at these questions?
Sure. So obviously the toolbox has changed, right, dramatically over the last 20, 30 years, especially with like now the optogenetic tools and the chemogenic tools that we have that allows us to manipulate these cell types very selectively. So we can activate them or inhibit them in a time restricted manner, and even in a projection specific manner. So we can look at what are the neuropeptides or neurotransmitters that these cells are releasing onto other downstream targets within their projection field. And this is also what we are doing. So this is what I'm also very interested in is understanding really how the communication of these hypothalamic cell types to others is happening, right? What is the whole purpose of this very complex communication system that we have? Why do we need, I mean, for example, why do we need a hunger and a satiety neuron? Why is not just a hunger neuron enough? And having just hunger turn on and off. Why do you need satiety neuron, right? Maybe a satiety neuron is doing even more. So this is where we had this idea that maybe satiety is not only satiety, but switches it to now eating something different, eating something sweet, right? Which you
Uh,
prefer instead of satiety.
uh,
So
yeah, that's very important given there's a lot of evidence that, that's taking a lot of sugar or high fructose corn syrup is a very popular thing because it's cheap for the agricultural. We got in the United States, there's cornfields all over the place. I grew up in southern Minnesota and went to college in Iowa, so that's like, the whole state is cornfields just about. And, some of that goes to feed uh, meat animals, but a lot of it goes to make this high fructose corn syrup, which, that ingesting that may have different effects on the circuitry than ingesting something that's like complex carbohydrates or whatever, protein or fat. So that's what you're getting at.
that's one thing, right? Like how is signaling coming from the gut, right? Like how is once you have it ingested, but then also the other question is what drives your behavior towards these things, right?
Yeah.
Why you're preferring something that is very sugar containing, very sweet. So I think there are two ways to look at it, right? One is what does it do to the body once you consume it, and then also to understand why you're actually consuming it and what are the mechanisms behind it that drive your behavior towards it.
Yeah. And okay, since we mentioned sugar, maybe we'll do this. I picked out a few of there's some really nice papers in top journals, and we'll go kind of in reverse chronological order if it's okay with you. Okay, so you had a paper in Science earlier this year, and it had to do with sugar appetite and these so-called POMC neurons, POMC neurons. Do you want to talk about that? And it also had to do with opioids. So there's, opioids, sugar appetite. So what's the connection?
So I think, I mean, this paper for us was, um, a really a great example of how we look at the circuits, right, and how we look at the cell types. Because what I said earlier that these palm C cells are very complex and difficult to understand because they're on the one hand producing this satiety signal, right, this alpha MSH, which is very, very famous. But then they're producing all kind of other neuropeptides, and we were always wondering, what are they doing? Why do we have them? And one is beta endorphin, right? So palm C cells are the only cell type in the brain that is producing beta endorphin. Beta endorphin is a very, very potent opioid
Yeah.
peptide.
Yeah.
So it binds with a high affinity to the mu-opioid receptor, and the mu-opioid receptor is very famous for its addictive properties, right? And like in that it is occupied or can be occupied by exogenous opioids such as morphine and heroin. And so we became really interested in understanding where and why POMC neurons are using this neuropeptide um, to influence anything. And we look back to older papers, and it was actually already described in the 80s, 70s, 90s, where people developed the first opioid antagonists. So, for example, naloxone or naltrexone. And they could show that in rodents, the blocking of endogenous opioid reduces appetite and particularly sugar appetite.
Hmm.
So this was for us then the link that we said maybe it's really this sugar appetite that home neurons are producing when they release beta endorphin. And then we thought to connect it as well as when we know that these neurons are active, and they're active in a state of where there is enough energy in the body. So after eating something or after having enough energy stored in your body. So in the state of caloric surplus, a lot of energy is there. So this is where we like put all of this together and like formulated the hypothesis. Maybe it's really the POMC cells that via the beta endorphin that they release is involved in controlling sugar appetite in the state of satiety.
Uh, okay, and then so these cells are producing beta endorphin. What cells are they releasing it onto to have these effects?
Yeah,
And
so
yeah,
so this is exactly where we then started, and we asked where in the brain could home C cells using this opioid, and so we looked histologically through the whole brain and found that there's an area which is the paraventricular thalamus or PVT
okay.
that is has really a high level of opioid receptor expression. So the cell types expressing it on the soma, and it had already been linked to um, withdrawal symptoms from exogenous opioids, so morphine withdrawal, and how rodents behave afterwards has been linked to this area. So that was very interesting for us, this like overlap of the projections from POMC cells and knowing that it causes or is some might be somehow related to motivated behavior, right? To motivate the animals to engage in a goal-directed behavior.
Hmm.
Yeah, and so then we started to look at it in different ways, right? Is it really beta endorphin signaling there? Do palm C cells at this site signal via beta endorphin? And we did multiple experiments to prove this. So we used electrophysiology with optogenetics. We used sensors to monitor opiate release. We monitored beta endorphin release itself. So really,
Wow. Yeah.
we try to like really do everything we can to make sure that it's beta endorphin there acting on mu receptors.
How long did it take you to do that? This was a lot of work. How many people were involved and how long from when you first thought of the general idea to when you actually got a paper submitted?
So I think it took quite long. So as you know, right? I mean, you do this experiment, and then one leads to another. Sometimes things lead nowhere.
Yeah.
So obviously, things take time. And this was, I guess, like 5, 6 years in total. It was one very, very talented postdoc I had in my lab, Marielle, and she recently left, and she was really, pushing this project in parallel to other two other projects that she was working on. So she has been, she's an extremely energetic person who run the show. And obviously, then we had collaborative partners who supported us in some experiments, but she was the one who was doing all of the main critical experiments to drive that project forward.
All right, so these neurons in the paraventricular hypothalamus. You also did another study you had in cell metabolism on this synaptic amplifier of hunger, and you found a, a, a neuropept well a peptide hormone. So I mentioned the hypothalamic pituitary system, and regulating the cortisol release, but there's also other releasing factors there, some that regulate the gonadal hormones and some that regulate the thyroid gland, which in this paper you found a role for the what initially I guess everyone thought was mainly function to regulate the thyroid gland is involved in this regulating hunger.
Yeah, so in this study we looked at the synaptic inputs onto AJP neurons and how these particular excitatory inputs can drive AJP neurons to control hunger. Because there were a previous study that suggested that uh, synaptic plasticity, driven by NMDA receptor activation, which is well known to be important in synaptic plasticity, that controls AJP neurons. And so, um, yeah, in this study, we found that there are these so-called TRH expressing neurons in the periventricular hypothalamus that are undergoing synaptic plasticity. But these cells, we don't think that they have anything to do with the neuroendocrine neurons. We think that they're also producing TRH, but they're not involved in the third hormone release itself because these cells are forming synapses onto AGAP neurons and to control hunger. And what this also very, extensive study showed was that these synapses between these yeah, TRH neurons and AJP neurons, they get amplified when you lose body weight. When the mice were either calorically restricted or fasted overnight, and that this we could causally link to a hunger drive, to increase in hunger, and then causing the body weight regain. So we think it's a very important mechanism that drives this body weight regain, which somehow was very interesting because it had it showed that it's not all driven by hormones that directly act on AGP neurons, but somehow there must be other pathways that come from potentially higher brain areas that coordinate also this hunger axis. And it's something that we're also still aiming to understand. Is there really maybe somewhere else in the brain or in the body and storage module for the body weight that organizes hunger in a way that if you lose body weight, it will always try to bring you back to your body weight.
Yeah.
Something that, is seen everywhere. Maybe, I guess most people have experienced this and they try to lose body weight that they at one point regain it, but it's also in the clinics, right? This is a really big issue for obesity treatment. Even with, uh, yeah, drugs such as the GLP-1 agonists, if you withdraw them, body weight regain occurs. So it's a really big issue because maintaining this lower body weight is extremely difficult. And we think that this hunger axis is so important for it because hunger is something that is very difficult to overcome, right? Once the hunger kicks in, that's this survival drive to consume, to not, lose too much energy to gain this body weight back.
So you mentioned GLP-1. So when I was at NIH, National Institute on Aging there, one of my colleagues, Josephine Egan, who's the head of the diabetes research group, she was working on, actually, she developed Xenon 4 or Xenotide, which is actually one of the earlier ones developed. And it's the one the peptide sequence is from the Gila monster. saliva. And initially the focus was on, fact that GLP-1 is enhancing insulin sensitivity, insulin release from the pancreas. And my take on this was early on, that was kind of the key mechanism for keeping blood glucose levels down, getting it out of the blood and into cells. But it seems to becoming much clearer that the benefits for people with obesity, maybe most of that's actually due to inhibiting appetite and even selectively, for example, you've done a lot of work on sugar appetite, maybe because there's this there's publications saying that the person's food preferences change. It's not just that they're eating less, they're eating less, if you will, obesogenic foods. But even then, the initial work, and that's still true, I guess, well, it turns out neurons all over the place in the brain have receptors for GLP-1. Hypothalamic neurons, but other regions. We did some work with Nigel Gregg, a colleague of mine, showing that it can have direct effects on neurons that may make them more resistant to stress. You had a really interesting paper. So the conventional thing is when you eat a meal, particularly if there's sugar in it, it causes release of GLP-1 into the blood from these certain of these cells lining the gut. And then the GLP-1 does things. It goes to the pancreas, causes insulin release. It goes to the hypothalamus. But you had a really interesting finding I wasn't aware of. Well, this is a little while ago now, I guess. On well, I won't spoil the take-home message, but you found that GLP-1s may be acting in an interesting way, affecting the brain by acting locally in the gut, not necessarily by being released into the blood and then circulating. Is that right?
Yes, I mean we have been studying the vagus nerve, right, as like
Yeah.
one way how signals can be transmitted from the gut to the brain. And one way is via the GLP-1 receptor, because as you said, right, the site where GLP-1 is released are these enteroendocrine cells, so that are lining the small intestine. And I think what's really interesting about GLP-1 is that it gets breaking down easily,
Yeah.
right, by enzymatic activity. And this is something why the amount that is released can have very profound local effects, but then it likely doesn't reach any organs far away, including the brain. So what we use is, so we develop different tools to be able to investigate the vagus nerve, to investigate the so-called sensory afferents of the vagus nerve, which transmits signals from the gut to the brain. And we also like investigated this GLP-1 receptor vagal afferents, and we found that fitting with this model that GLP-1 is a signal that uh, signals now nutrients come in, and you should eat less. That when we activated them, we saw a reduction in food intake, but we also saw that that improved glucose tolerance. So we think that this is another way how communication between the gut and the brain can be a way how glucose metabolism can be improved, right? And how that neuronal transmission is very important.
How did you look at it? You know, so far you've talked about actually looking at the neurons in the brain with these, modern techniques where you can look at their activity, figure out which transmitters are involved and manipulate those in ways that affect hunger and satiety. But so did you, I guess I miss, I don't know if I got this from the paper, but did you actually look at uh, somehow block GLP-1 receptors locally on the vagal sensory neurons? Because that's kind of a key thing to show it's acting locally, right?
No, that would have been a key thing. So this is not what we did. So we
Ah.
we manipulated them chemogenetically. So we targeted them with a genetic toolbox.
Mm.
What we did not look at it in a receptor specific manner. So that is something you would need to do to really tease apart where the receptor acts. And I don't think anyone has done that. I think that is very, very challenging because I
Yeah.
mean you need to affect the protein, the receptor, and I don't think anyone even knows where the receptor is at, right? Where peripherally speaking, the protein is yeah being there for sensing the GLP-1 that is released. So that's definitely an area of huge interest.
So these neurons that are projecting to the gut through the vagus nerve, are there cell bodies in the brainstem? They're not in the hypothalamus, right? They're in the brainstem?
So the cell bodies are in the nodose ganglia. So the nodose ganglion, they that's the cell bodies were of the vagal sensory neurons, and so they have terminals that are the peripheral terminals that are innervating the organs, and then their central terminals projecting to the brainstem, particularly to the NTS, so the nucleus of the tractus solitarius, and the area postrema. So they're transmitting their information, they're detecting in the peripheral organs, so the eye tract, but also other pancreas, lung, and then transmitting this information to the brainstem.
Okay, but then is there connections between the brainstem and the hypothalamus that are, in other words, if you have GLP-1 acting receptor way down in the gut, is that leading to effects in the AGRP or POMC neurons that affect hunger, or is this something independent?
So it's definitely there's a bit of a black box, right? What is getting to the brainstem, and then what is getting to the hypothalamus. We know there must be connections. Because when I mean, there's a very neat studies to demonstrate when you manipulate vagal sensory neurons, you can monitor changes in AGFP cells, and they demonstrate, right, there must be communication. But how exactly this is wired, that is still kind of a black box. So it's something also we're aiming to determine. Um, the other question obviously is how do these exogenous GLP-1 agonists then work, right? Where they have been changed in a way that they now stick around for much longer and they can enter the brain and can work basically everywhere where the GLP-1 receptor is located. And that includes the hypothalamus. As well as many other brain areas, as you said, right? So that is a complete different level where you not only have the endogenous action of the GLP-1 working locally in the peripheral organs, so close by to the intestine, but now you flood the whole system with an agonist that affects a lot of cell types expressing the GLP-1 receptor.
Yeah, it's interesting. There's so many trials going on with these GLP-1, long-acting GLP-1 receptor agonists and all sorts of uh, uh, obesity, diabetes definitely benefit. And then there's going to be secondary benefits, at least in the short term, for the cardiovascular system, pretty much anything, anything that obesity causes. But in the area of neurodegenerative disorders that I worked on a lot, Alzheimer's, Parkinson's, there's a trial the trials in Parkinson's, look favorable in the first ones, but then a phase three, a big study, there didn't seem to be a benefit in slowing the decline. It's not clear with Alzheimer's. And so I actually had a colleague at NIA, Dimitros Kapagiannis, that did a small trial of exendin 4 in patients with early Alzheimer's disease, and he didn't see any benefit. But these actions elsewhere, we really don't know what the heck's going on there. So it'll be interesting when people start looking at, you know, long-term effects on whatever mood, learning and memory, and then try to tease out, whether there's effects of GLP-1 on the circuitry that are independent of an indirect effect, say on whatever um, brain health, uh, indirect just because the person's losing weight, and we know that's good for the brain. Yeah.
No, absolutely. And I think this is still right. I mean, it's a very potent drug that works extremely well for obesity. And then it has all these additional effects. And I think we are not even understanding how it can be so effective for obesity treatment. And then on top of it, like what are all the other effects it has and where it comes from. And so
Yeah.
yeah, I find it fascinating that it has so many positive effects. And but it's still such a black box. And there are also these studies now that show when you take out some of these cells, right, in the brainstem or in the hypothalamus, that are that have the GLP-1 receptor,
Uh,
then suddenly the system doesn't function anymore. Then the drug doesn't efficiently reduce body weight anymore. And it's so interesting if you consider, right, why do you need all of them, or which of them are actually the ones that are very important? So that is, I think, where we're still lacking in understanding how this drug works.
yeah, something I mentioned before we started recording is that, just in kind of basic endocrinology, neuroendocrinology, is you have a hormone. I guess we can call GLP-1 a hormone or paracrine factor, whatever. And they're released in response to some physiological stimulus, in this case, eating, right? But uh, cortisol is released in response to stress. Thyroid hormone responds to and controls energy metabolism, etc. Uh, and so generally, my thinking was it's not good to have some hormone at abnormally high levels chronically. Seems like something bad is going to happen in the long run, but it's so far that seemed quite safe without major long-term side effects. So what's your thinking on why that would be? In other words, so the leptin, you mentioned leptin resistance, right? Where the hypothalamic neurons don't respond so well to leptin. It's true in people with obesity, for example. Um, and same with insulin resistance. You can have very high levels. The pancreas starts cranking out a lot of insulin. So typically people with type 2 diabetes chronic have elevated insulin levels because the system's trying to compensate somehow for the failure of the cells to respond appropriately to insulin or leptin.
Yeah, it's a very interesting question. I mean, it's always difficult to say what is resistant and what not, right? Because it's the insulin resistance is different to what people refer to as the leptin resistant. There's a big debate in the field if that is really a resistance. But anyway, right, the drug or the hormone doesn't work anymore as efficient as it should, right? So leptin in obese patients can, when given exogenously, no longer produce its effect because the system is saturated, right? There is so much leptin there, the receptor can no longer be activated. So I don't know why the GLP-1 receptor works in a way. I mean, I think it's if you look at it, how much the GLP-1 agonist can do, it's also there seem to be also a resistant, right? Or like it levels out, right? Because the weight loss in patients is 15, 18% 20, right? This in average. But then you can get a further body weight reduction with semaglutide itself. And so is that then a new state that you're in, that you basically have the system again in a state of resistance, or if you want to call it this way, that now it is saturated and you cannot go beyond that. And that obviously the back to the question, like is that now the physiological, right, where you in a healthy environment you would have that, and that is just what you're with the drug can reinstate. I don't know. I mean, that's a big question mark there.
What are your what are your questions you're trying to address now in your current research? The outstanding questions. What's your lab focused on now?
So we continue doing a lot of looking at the synaptic protein, synaptic changes, synapses themselves. I think it gets back to what you said before, right? People looked at the hypothalamus as like a neuroendocrine part of the brain. And the more we learn, or the more I learn, the more fascinated I am that this brain part seems to be very different in how it communicates, how the synapses can also undergo plastic changes. And maybe, or I think that this is maybe where something very important can happen to the system and how the system can adjust to environmental factors, how to internal changes, and maybe that is where also these effects occur. How why obesity can know or is very difficult to bring back to a normal body weight, right? Where over a prolonged period of time, these synapses become activated or silenced, and this is really what happens. Where yeah, it's the going back from the higher body weight is so difficult. And so this is really where I have a lot of interest in where we're going in the hypothalamus, but then also from a brainstem uh, perspective, how that information gets to the hypothalamus. So
And
really about the
do you have any plans or ongoing collaborations with clinical people at any interest in doing, translational work?
yes, we're always very interested in, on the one hand, determining whether whatever we found is important for humans as well. I think it's if not, that's also fine, right? That's basic knowledge. But obviously, for the health of humans, it's very important to understand if the cell types and the mechanisms that we find also exist in humans, and then also to use our knowledge to develop maybe new ways how we can target the system, maybe in a different way, maybe in a more targeted and a more specific way that allows us also to, on this synaptic plasticity level, work and have effects, maybe reverse plastic changes that are rather pathological.
Yeah, you mentioned there's certain uh, I guess clinical examples where someone has a gene variant or mutation that causes overeating. And what's going on as far as uh, so you can have a gene where a mutation, it's going to cause severe overeating. Um, but you could also imagine more subtle genetic alterations that increase your propensity to overeat. For example, maybe overeat sugar or overeat fat or something like that. Um, and then I guess you could, if if there are such genetic variants being identified, then you could play around in the mice, manipulate those genes.
Yes, absolutely. I think yeah, the melanocortin pathway obviously has already been identified,
Yeah.
and there's still a big question of like how many people are affected by it, because we learn more about that really even small defects in the receptor or its expression level can cause
Hmm.
an increase in food intake. And um, that, as we know, right, even or just eating a little bit more than you need per day can over time cause obesity. So that is actually very important, right?
Yeah.
Especially in our environment these days, but we have an unlimited access to food and palatable food. And I also think that there are differences, right, that can be traced back to genetics that why sometimes drug work better or less good, right? Like these GLP-1 agonists. And some people they have more severe side effects or are more responding to it versus others. And combination of different drugs might be preferable, right? Based on genetic differences. So
Hmm.
um, yeah, that's where we're also trying to by getting a really basic understanding of how the system is working, maybe we can explain the things, but then also translate it to direct it better and get new ideas about it.
Okay, great. Well, uh, I don't know if you can hear this noise that I'm hearing outside here, but there's some company cutting branches off trees and grinding them up. But I think my Mac actually is pretty interesting. I think somehow it screens that noise out pretty good. It kind of, I don't know if there's artificial intelligence, but it seems like the computer knows what's, a person talking versus.
I
uh,
I don't hear anything.
okay. All right. That's great. All right. Well, thanks a lot, Henning. I really appreciate you taking the time, and I'll let you get back to work with your lab.
Thanks very much. It was a pleasure talking to you.
All right. Bye.
Bye.